Milk is an emulsion or colloid of butterfat globules within a water-based fluid that contains dissolved carbohydrates and protein aggregates with minerals because it is produced as a food source for the young. All of its contents provide benefits for growth. The principal requirements are energy, lipids, lactose, and protein, biosynthesis of non-essential amino acids supplied by proteins, essential amino acids and amino groups, essential fatty acids, vitamins, and inorganic elements, and water. The pH of milk ranges from 6.4 to 6.8 and it changes over time. Milk from other bovines and non-bovine mammals varies in composition, but has a similar pH. Initially, milk fat is secreted in the form of a fat globule surrounded by a membrane. Each fat globule is composed almost entirely of triacylglycerols and is surrounded by a membrane consisting of complex lipids such as phospholipids, along with proteins. These act as emulsifiers which keep the individual globules from coalescing and protect the contents of these globules from various enzymes in the fluid portion of the milk. Although 97–98% of lipids are triacylglycerols, small amounts of D and monoacylglycerols, free cholesterol and cholesterol esters, free fatty acids, and phospholipids are also present. Unlike protein and carbohydrates, fat composition in milk varies widely in the composition due to genetic, lactational, and nutritional factor difference between different species. Like composition, fat globules vary in size from less than 0.2 to about 15 micrometers in diameter between different species. Diameter may also vary between animals within a species and at different times within a milking of a single animal. In unhomogenized cow's milk, the fat globules have an average diameter of 2 to 4 micrometers and with homogenization, average around 0.4 micrometers. The fat-soluble vitamins A, D, E, and K along with essential fatty acids such as linoleic and linolenic acid are found within the milk fat portion of the milk. Normal bovine milk contains 30–35 grams of protein per liter, of which about 80% is arranged in casein micelles. Total proteins in milk represent 3.2% of its composition. The largest structures in the fluid portion of the milk are casein micelles, aggregates of several thousand protein molecules with superficial resemblance to a surfactant micelle, bonded with the help of nanometer-scale particles of calcium phosphate. Each casein micelle is roughly spherical and about 1/10 of a micrometer across. There are 4 different types of casein proteins, S1, S2, and caseins. Collectively, they make up around 76–86% of the protein in milk, by weight. Most of the casein proteins are bound into the micelles. There are several competing theories regarding the precise structure of the micelles, but they share one important feature: the outermost layer consists of strands of one type of protein, K-casein, reaching out from the body of the micelle into the surrounding fluid. These kappa casein molecules all have a negative electrical charge and therefore repel each other, keeping the micelles separated under normal conditions and in a stable colloidal suspension in the water-based surrounding fluid. Milk contains dozens of other types of proteins beside caseins and including enzymes. These other proteins are more water-soluble than caseins and do not form larger structures. Because the proteins remain suspended in whey remaining when caseins coagulate into curds, they are collectively known as whey proteins. Whey proteins make up approximately 20% of the protein in milk by weight. Lactoglobulin is the most common whey protein by a large margin. Minerals or milk salts, are traditional names for a variety of cations and anions within bovine milk. Calcium, phosphate, magnesium, sodium, potassium, citrate, and chloride are all included as minerals, and they typically occur at concentration of 540 mm. The milk salts strongly interact with casein, most notably calcium phosphate. It is present in excess and often, much greater excess of solubility of solid calcium phosphate. In addition to calcium, milk is a good source of many other vitamins. Vitamins A, B6, B12, C, D, K, E, thiamine, niacin, biotin, riboflavin, folates, and pantothenic acid are all present in milk. For many years the most accepted theory of the structure of a micelle was that it was composed of spherical casein aggregates, called submicelles, that were held together by calcium phosphate linkages. However, there are two recent models of the casein micelle that refute the distinct micellular structures within the micelle. The first theory, attributed to de Kruif and Holt, proposes that nanoclusters of calcium phosphate and the phosphopeptide fraction of beta-casein are the centerpiece to micellular structure. Specifically, in this view, unstructured proteins organize around the calcium phosphate, giving rise to their structure, and thus no specific structure is formed. The second theory, proposed by Horn, the growth of calcium phosphate nanoclusters begins the process of micelle formation but is limited by binding phosphopeptide loop regions of the caseins. Once bound, protein-protein interactions are formed and polymerization occurs, in which K-casein is used as an end cap, to form micelles with trapped calcium phosphate nanoclusters. Some sources indicate that the trapped calcium phosphate is in the form of Ca9, PO4, 6, whereas others say it is similar to the structure of the mineral brushite CaHPO4, 2H2O. Milk contains several different carbohydrates including lactose, glucose, galactose, and other oligosaccharides. The lactose gives milk its sweet taste and contributes approximately 40% of whole cow's milk's calories. Lactose is a disaccharide composite of two simple sugars, glucose and galactose. Bovine milk averages 4.8% anhydrous lactose, which amounts to about 50% of the total solids of skimmed milk. Levels of lactose are dependent upon the type of milk, as other carbohydrates can be present at higher concentrations than lactose in milk's other components found in raw cow's milk are living white blood cells, mammary gland cells, various bacteria, and a large number of active enzymes, both the fat globules and the smaller casein micelles, which are just large enough to deflect light, contribute to the opaque white color of milk. The fat globules contain some yellow-orange carotene, enough in some breeds, such as Guernsey and Jersey cattle, to impart a golden or creamy hue to a glass of milk. The riboflavin in the whey portion of milk has a greenish color, which sometimes can be discerned in skimmed milk or whey products. Fat-free skimmed milk has only the casein micelles to scatter light, and they tend to scatter shorter wavelength blue light more than they do red, giving skimmed milk a bluish tint.